The genome annotation job at BV-BRC brings you a lot of really fabulous gifts. First, there are all these different files and formats of data that come with your annotated genome. There's this genome report that it gives an indication of the quality of your genome and also potential problems that you might have with it. But perhaps the greatest gift is that your private data is virtually integrated with the rest of the resource. Now, only you can see it, but it'll look like everything else. And once you have this annotation job, you can use a number of different services to go deeper into the analysis of this genome that you isolated in the lab, perhaps, and paid for the sequencing. And now here it is. So we're going to finally look up here at these three icons that are hyperlinks to your private data. One is the genome landing page. One is the feature page for all the genes in your genome. And one is the genome browser. I always first, pretty much exclusively, click on View Annotated Genome. So you click on that and it rewrites the page to show your particular genome. Now notice that there's a lot of information here on this first page. At the top, you can see the breadcrumb, but it's also the taxonomy of your genome. It's showing you the species, the genus, the family, all the way up the phylogeny. If you click on these, it's going to go specifically to that taxa. There's a lot of information here. A lot of it we saw in the job results, but here you have a breakdown of the proteins. You have information about the phylogeny. You have our unique genome identifier. And if you scroll down, there's more things that it has about what's annotated on the genome. And some of these things were things that you saw in that genome report. Now, remember way back when we talked about the annotation pipeline, and I told you that we blast every gene or protein in the genome against the specialty gene database. These are the hits to that. And you can click on those and see what they are. And then you can see the protein families that have been assigned. And you can see the number of genes, tRNAs, and rRNAs. And when you look here and you see GenBank or RefSeq, those are all 0. Why is that? Because it's your private data. We didn't get it from GenBank. We didn't get it from RefSeq. This is yours. This is your data, so this will be blank. So there's all this information. And one thing I want to point out is if you scroll down here and you see that there's no phenotype or host information, you could add that by going up here and clicking on edit, and you could enter in data that you wanted to put in. Let's see if I can find one for the isolate. It's human. And I could put that in, save it. I don't think we'll see it right away. It takes a while for that data to be updated. Oh, see, there it is. I lied, it's right there. So you can edit the metadata there, and then it would be available when you look at genomes across the resource, and let's say you were filtering on those isolated from human, your private data would be there. So there's all this stuff. But look, there are also all these extra tabs that we have of things. Now, some of these, because it's private data, you won't see those things like protein structures. This is your private genome. We're not going to have protein structures. We will have pathways and subsystems, but we're going to delve more deeply into those when we do comparative systems. There'll be no experiments with this or interactions. A lot of this is for public data, but I'm going to show you the stuff that we have private data for. So AMR phenotypes, okay. If it's a different genome, remember when I went over those lists that it started off with Acinetobacter baumannii, and it had Streptococcus pneumoniae, Staph aureus, those particular genomes, when you click on this tab, it'll tell you what the predicted antimicrobial resistance is for this genome. We don't have this for Haemophilus influenzae. For phylogeny, it should just be a tree of Haemophilus. This looks pretty nice, and some outgroup information here. Genome browser is your genome, and it will show all the contigs. And like I can click on this, you can see there were 32 contigs. So I should see them all here. And you can zoom in here, keep zooming. You won't see the RefSeq annotation because this is a private genome, but when it's a public one, you'll be able to see that. And then you'll be able to get into the 6' translation. And if you have something that you think is potentially a pseudogene, you can look for the problems here. The circular viewer is showing each of these as a contig. So this is one big contig. So you can see these are the contig barriers here where these little white spots are. These are the different layers that are in the circular viewer. I often see this particular viewer in publications that are citing the resource, so people take this. And it's also pretty cool because you can adjust it. For example, if I wanted the virulence factors to be magenta, I can do that, and it should change those to that particular color. So you can manipulate it. Let's look at sequences. These are the contig sequences. This is the only place that you can get this unless you're doing the command line interface. So it's showing you all the different contigs and their length and GC size. And if I wanted an individual one, like let's say I wanted to blast it against something else, I could download it here. Features are all the genes. And down here, it tells me how many genes are annotated in this genome. If I click on the features, you can see that I could filter on tRNAs or coding sequences, and it will automatically filter the table to show that. And if I wanted all of the coding sequences in a particular file, I could select them all and I could download it. I could get the DNA FASTA, the protein FASTA, or I could get it as a text file or a comma-separated value file. Also, if you just want a single one, if I click on a single one, it gives me information about that gene. Here, it tells me the protein families and everything else. And were I to click on the feature tab, it would take me to the landing page for that particular gene. So let's talk about the proteins. It's just a different flavor of what we saw in the feature table, and these are the particular proteins. And then the specialty genes. These are the ones that you blasted against a specific database. This happened during the annotation. It's a free gift to you. So let's click on the filters, okay? And you can see you can filter on the property. These were the only ones that they had hits to. So let's say I'm interested in antibiotic resistance genes that came from the annotators that are associated with the project. And here they would be, and look, oh, now this is pretty valuable. So it also shows you that this was a k-mer search, and that's how these genes are identified by a k-mer function here. So I click on this, and notice here it's got a publication associated with it. So if I click on this publication is a hyperlink. So let's click on that, and it's going to open up a new tab, and it's going to show me information about that particular gene is somewhere in this article. So finally, we have the protein family. So let's click on that. And this is the protein family table. And sometimes it takes a few minutes to load, but it's showing me all the genes that are in protein families in this genome. And you can also see it as a heat map. If you're interested, you can scroll it out to see what those genes are, the ones that have 2 or more copies. And generally, I mean, we've got scrolls all over the place here because it also scrolls in this direction. You could filter on keywords. I don't really care about hypotheticals, but generally I spell that pretty well. So I'll go back to the table and filter on hypothetical. So it's got 166 families. And if you wanted to look at those in the heat map, this is what they would look like. We could go into pathways and subsystems. It takes a while to load that to go into detail on these two things. We'll do that when we go over comparative systems, but I just wanted to show you briefly that I can choose a pathway and I can say I want to see the map. And we're waiting for it to load. This is a KEGG map. And these are the genes in the light green that are annotated in my genome. So how cool is that? You have all this information at your fingertips. So start annotating your stuff so that you can look at this too. See you later. Bye.